is a pleasure to have for this special lecture Antonio Vidal, or Tony Vidal Puig. We know each other for a number of years. He was born in Valencia, studied medicine there, and then did his residency and medical practice in the south of Spain, in Granada, and then moved to do molecular biology. I learned about molecular biology and nutrition, endocrinology. He's an endocrinologist, I think, by training. And he moved to Harvard, where he stayed there till, I think it was 1999. And there he was started working on molecular cellular biology, also manipulation of animals, so creating knockouts, transgenic mice. He had done a wonderful work on manipulation of animal models on energy metabolism. And then moved to Cambridge, where he had a successful career. I think 2 years ago was promoted to be professor of Cambridge University at the Department of Molecular Nutrition and Metabolism at Cambridge University. It's probably, well, I think it's the first time a Spanish educated, I mean, somebody from Spain that got a degree in Spain became professor at Cambridge, which is probably one of the universities that has more Nobel Prizes per staff. So it's,
I don't see where you're going with this.
Well, you're going to talk about adipose tissue, BMP, well, and about his talk. I think we are very fortunate that the title of his talk, BMPs and thermoregulation, is totally different than what you saw in adipose tissue expandability. The reason is that we're going to be fortunate. It's the first time he's going to talk about this. Last week got accepted a paper in Cell, and he wants to share with us all the information of this paper. If you like the paper, you get a good lunch. Thank you for being here.
Okay, thanks for the invitation to be here. And yesterday I gave my presentation to the lay public, which I really enjoy, and I think I gave them the messages for lay public and healthy lifestyles. And today I need to put another talk, and I was tempted to put the one that I've been presenting over the last few months, but I thought there was an opportunity to put together the presentation with our new staff and present it here. I didn't know that I was going to be recorded, so I just make sure that we don't get this exposed until the paper is published. And basically, my program of research focuses on obesity, and as you know, in obesity research there are two key questions. One of them is why people get obese, and this is a question that needs to be addressed from the point of view of the mechanisms that control food intake and energy expenditure. In order to be obese, you need to have positive energy balance. That's not the key question that we are addressing, although sometimes, I think the second question, which is I think relevant, is why being obese causes metabolic problems. My laboratory traditionally has been focused on the second question. What happens is that when you start looking for the second question, sometimes you come out with aspects of it that are very relevant to the first question. So, and being pragmatic is not because you choose one question, you should not address the other one and you come across something that is interesting. My laboratory has an adipocentric point of view of the metabolic syndrome. A syndrome means that you can get into specific pathological entity through different entrances. You talk to people working in metabolic syndrome, you may get different flavors. Some people may suggest that the key organ for the metabolic syndrome is the muscle, because it's the key organ that will uptake glucose, and then that's the entrance to the metabolic syndrome. Other people may think it's the liver, because the liver produces a lot of glucose that can get into blood, and that is fundamental for the metabolic syndrome. Other people may think it's in the brain, and probably they may be true on this. But we think that the majority of people with metabolic syndrome are obese. And from this point of view, we think that the adipose tissue, which is the common feature to all these people, may be central to their problem. So we think that the link between obesity and the metabolic complications is related to a mismatch between energy availability and storage capacity. And we think that the key word for this is lipotoxicity, in the sense that we think that the excess of nutrients that cannot be accommodated in the adipose tissue tend to be accumulated in other organs where they cause toxicity. And in some way, if I were going to rename the metabolic syndrome, I may rename it as the overnutrition toxic syndrome, in the sense that we think that at the root of the problem is overnutrition. So from this point of view, we think that the metabolic syndrome is the combination of an environmental problem that results in an increase of nutrient availability and a genetic component that is determinant of the genetic makeup of specific organs. And this genetic makeup is what gives the specificity to the phenotype. Some people develop obesity, others develop diabetes, others develop heart failure, others develop liver problems. And we think that it's in the genetics of these organs that determines the resilience of the organ to the insult and whether they can cope with the excess of nutrients there. So in some way, it will be a common insult, and the vulnerability of specific organs what determines the symptomatology. From this point of view, our program of research is focused on the question of the link between obesity, insulin resistance, and cardiometabolic complications. Our hypothesis is that the failure of the adipose tissue, expandability and function results in lipotoxicity, and we define lipotoxicity as the ectopic accumulation of lipids in organs different from the adipose tissue, and we believe that this is important in linking obesity with the metabolic complications. From this point of view, our research has a common thing, which is lipotoxicity. And from this, we have three main hypotheses that drive our program of research. We do things that apparently are different, and people sometimes say, well, you know, work in A and B and in C. But all of these projects can be integrated in one of these three hypotheses. The first one is, if we improve the capacity of the adipose tissue to store fat efficiently, this is going to have a knock-on effect preventing insulin resistance. So that means that we want to understand how the adipose tissue develops, how cells get recruited to become adipocytes, the difference between hypertrophy and hyperplasia, and how this impacts in the metabolic syndrome. The second hypothesis, which is relevant to what I'm going to be telling you today, is the concept that if your adipose tissue fails, the second factor that is going to be determinant to help you not to become diabetic is your oxidative capacity. If we say that the problem is that the lipids get in other organs and cause problems, any strategy that will clean up these organs, any strategy that will help to remove the excess of nutrient in these organs may contribute to prevent the development of metabolic complications. And over the years, we have used several strategies. We have used mitochondria and coupling as a strategy to try to burn more in an efficient way. And we have considered exercise as a strategy to increase mitochondrial units and increase oxidative capacity. And more recently, we have considered the possibility of transforming or trying to focus in aspects that will transform white adipose tissue into brown adipose tissue. And considering brown adipose tissue as an organ able to oxidize excess of nutrients that helps them in some way to prevent storage in organs like liver, muscle, and so on. So if we can make people to develop more brown fat, or we can make people to make their brown fat more active, that will act as a sink for glucose and lipids and will clean up other organs, and in some way may prevent metabolic complications, and in some way also may prevent the development of obesity itself. The third hypothesis that we will not address today, but I think for the sake of completion I would like to mention here, is the concept that if your adipose tissue fails, you don't have oxidative capacity that is appropriate, the third factor that is going to determine whether you develop metabolic complications or not is going to be the quality of the lipids. We know that there are some lipids that tend to go to specific pathways that are going to be more or less toxic. We know that accumulation of triglycerides is a defensive mechanism that allows the system to store fat efficiently in a non-harmful way. And that failure to do so results in lipids going other pathways that are a little more toxic that can accelerate some of these type of insulin resistance and associated complications. So in some way, if we devise strategies that help to prioritize specific pathways that are relatively less toxic, we can increase the buffer capacity of these organs to delay the progression of the metabolic complications. So if you are 55 and we can give you 10 years of delay in the metabolic complications, that is important. Having said that, I will focus now in the concept of the brown adipose tissue. I don't know how much you have been exposed to the, information about this organ, but there are two different types of adipose tissue, the white and the brown. The white main function is to store fat for the purpose of storing energy to make it available when it's required by other organs. The brown fat is an adipose tissue in the sense that they have fat inside, but it's very clear that this organ main function is not the storage. The main function of this organ is to make heat. So in this regard, it's very relevant in small mammals, it's very relevant in newborn because they have a large surface compared to the volume and they are dissipating a lot of heat, so they need to make heat. And this type of organ allows to transform chemical energy into heat without making work. So in some way, it's a waste of energy making that helps to maintain temperature. So you want to store fat, obviously you have characteristics of the adipose tissue that allows exactly this. So this white adipose tissue has mitochondria, but just to allow the adipocyte to survive because they need energy. However, the brown fat is an adipocyte which is full of mitochondria. And these mitochondria are not normal mitochondria. It's not like mitochondria in your heart or mitochondria in your muscle. These mitochondria, they have specific transporters that are uncoupling protein 1. And this type of transporter, what it does is to uncouple the oxidation of fatty acids from energy production. And what they make is heat. Basically, it's a hole in the membrane. So you look at this figure, basically, okay, this is the white adipocytes. These are the mitochondria that appear in the remit close to the membrane. This is how a brown fat look like. These are small little droplets within the cells. And if you look at the electron microscopy, what you have is tons of mitochondria there, which main function is to make heat. So, and the idea is you have more of this, that will make you to need less of that, and that will make you healthier. So why is interesting the brown fat? Okay, for all the studies that you do in rodents, whenever you want to understand obesity and diabetes, whenever you try to develop treatments that you want to test in animals, you need to consider the brown fat. Because the brown fat by itself can use more amount of glucose per volume unit than the muscle. The brown fat by itself can contribute to metabolism. And for example, you can have a mutant in a mouse, and you put the animal at room temperature, or at thermoneutrality, 30 degrees, where the brown fat is not working. And then this mouse model may have diabetes and obesity, as you may expect, okay? But you put this animal at a lower temperature, where the brown fat will become activated. And a mutant in a human being produces a very severe phenotype will disappear in the mouse, because the problems that you have of insulin resistance are totally bypassed by the brown fat that is taking glucose inside. And this is independent of the action of insulin. To the point, for example, that I know there's a digression here. There was one patient in the States, she had a defect in the insulin receptor. She was diabetic, 1,000 units of insulin, all the different drugs that you could utilize to try to control her diabetes. And this person developed a tumor in the thyroid that resulted in removing the thyroid gland. And they put her on replacement with thyroid hormone. And they put a dose that was slightly higher than normal because they wanted to prevent the tumor from coming back. In 2 years, this person was progressively decreasing the amount of insulin that was required to maintain their control of their diabetes. And at some point, this person, despite having a mutant in the insulin receptor, didn't require more insulin to treat their diabetes. What this person had developed in the back was a big fat pad of brown fat that actually was sucking the glucose and was making insulin unnecessary. Even now, there are some suggestions that you can have a type 1 diabetic rodent. If you put them in the cold or you activate the brown fat, you may even bypass in some way the need for insulin in the model, and they may become normal glycemic. So what I'm saying is, it's a small organ, but extremely powerful. Relatively small volume of this organ can really have an impact in metabolic control. The second thing is that in humans, I mean, until now, working in brown fat was something that was limited to small laboratories lost in the middle of nowhere because people thought that it was irrelevant. But for us, everything changed a couple of years ago when important people decided that that could be important, and they published papers in top journals, and suddenly the topic became relevant. And then we could capitalize on the subject that we have been working without hopeless for many years, now become relevant. So that means this seems to be that brown fat is not only relevant in children, but actually adults have some brown fat. Adults can induce the differentiation of brown fat. And in fact, if you compare lean and obese people, the lean people tend to have more brown fat than the obese one, which opens the question of whether it's chicken or egg. Because we don't know if they are obese because they don't have brown fat, or because they are obese and they are thermoinsulated, they don't have brown fat. Okay? So the other concept I would like to put forward is how we decided to work in the molecule that we are interested in. There is a philosophical question behind this. Many of our animals, when we introduce specific mutants that are like PPAR gamma 2 knockout mutant, we were expecting a lipodystrophic, insulin-resistant, diabetic animal, and we were expecting the adipose tissue to collapse in this animal model. However, our PPAR gamma 2 knockout mouse in a 129 background was remarkably inappropriately healthy. And then for many people, when they get a remarkably normal, healthy animal, despite expecting a very severe phenotype, they think the animal is not interesting. However, we ask the question, why my mouse is normal? I'm not expecting this animal to be normal. So this animal either is not challenged enough, or B, they have some alternative adaptive mechanism that the animal can activate to try to make, to remain healthy for as long as possible. So in our animals, there was something interesting, is that we could take preadipocytes from the white adipose tissue, try to differentiate them in vitro, or we could take adipocytes from the preadipocytes from the brown fat. In the animal, the adipose tissue looked normal, but we couldn't differentiate them in vitro. So there was something that was intrinsic to the tissue that was defective, but in vivo, the animal managed to overcome the problem. So that means something in vivo, and that, from the philosophical point of view, means that the animals will do whatever they can to try to minimize the effect of your genetic perturbation. And we think that if we focus in this adaptive mechanism that the animal uses for this, we may identify two things. Number one is we could identify markers of stress, because anything that tries to compensate, you could identify as a reaction to a level of stress that you need to compensate. That's a marker that could be useful. The other thing is, yesterday I was saying to people, okay, losing weight is very difficult, because you do something and the body will try to compensate to remain or to maintain energy balance. In some way, you want to target the specific treatments for obesity, you need to identify not only the effectors of the energy balance, but you want to identify the different points of control that are going to determine adaptive mechanisms that can prevent the success of the intervention. So from this point of view, we did some profiling of the brown fat of these animals. And the molecule that, at that moment we were really bad in the analysis of the data. So the pathway analysis at that time was not something that we were very good at. And we ended up with the top 10 candidates that we wanted to follow. And then we found out this BMP8 beta, or B, and we thought it could be interesting from the point of view that was a bone morphogenic protein, the fact that this could contribute to the differentiation of cells. We know other bone morphogenic proteins, this is what they do, they contribute to differentiation. And we thought that maybe the animal is surviving because in the absence of PPAR gamma, then they somehow activate more differentiation by alternative mechanisms. So the concept of the bone morphogenic proteins, we know that they belong to the family of the TGFs. They know that they bind the type 1 and type 2 receptors that dimerize, and then this results in activation of SMADs. There are some of them that are regulatory. They translocate to the nuclei, and that defines some specific gene transcription pattern. The other thing is that we know that there are alternative signaling pathways that contribute to the transcription, and particularly to the brown fat that are coming through the PI3 kinase, and particularly through the MAP kinase, kinase 3, 6, and the one that appears in all our models, which is the P38 MAP kinase. So we decided to try to follow this lead. The previous information about the other BMPs suggested that they received the name of bone morphogenic protein because obviously some of them have something to do with bone formation. But we found that BMP4, for example, has something to do with preadipocytes, the differentiation. When we started with BMP8, we didn't know anything about BMP7. That is an interesting experience. We put grants to work in BMP8, and we never got any money because no one thought that BMP could be relevant for this. And then when the other guys published about BMP7, in some way for us was negative, but in some way for us was positive, because after this, the reviewers changed their mind. I thought, maybe these guys are up to something really good. The conclusion from this is, it's good to be a pioneer, but it's very hard. And sometimes you need to get another one who is a very senior one, more money, they get a hint, and then you should not take this as a problem. Sometimes this facilitates you getting the money and maybe showing that the other guy was wrong, which is okay. So we start to look at BMP8 and B, and then if there is a BMP8B, it means that there is a BMP8A. And we start to look at the tissue distribution of both of them. As many other things that we have been working in thermogenesis, they are very highly expressed in the testes, and I don't know why, but I guess it's a lot of mitochondria are there. And then we found that the BMP8 beta was very highly expressed and very specifically in the brown adipose tissue, whereas the BMP8A was not there. In the white adipose tissue, predominantly was BMP8A, but was substantial of BMP8 beta, much less than in the brown fat. And the other place that was interesting was that the BMP8 beta was also in the brain. Okay, so it was brown fat and brain. And in the other organs, as important as that BMP8 beta was in the right places for us, was that the BMP8 beta was not in the other places that we didn't think was interesting for us. The other interesting thing about this is, if you compare the stromovascular fraction with the mature adipocyte, you will expect that the BMPs will come predominantly from the stromovascular fraction, because if they contribute to the differentiation, it's more likely that you will find them early on in the stromovascular and then contribute to the process of differentiation. And this is what we found with respect to all the BMPs. All the BMPs, 2, 4, 5, 6, even 7. 7 has been shown that contributes to the differentiation of the brown fat. However, our BMP8 appeared in the mature adipocytes. So in some way, we were looking for a molecule that a priori we thought was going to be relevant for differentiation, but it appeared mainly in an adipocyte that was brown and extremely well differentiated. This is not something that you could expect from a bone morphogenic protein with respect to the usual role. The other thing that was interesting is the pattern of expression during the differentiation of the brown adipocytes. So BMP8B was not present day 0, day 3, day 6, day 9. It was highly induced very later on in parallel with UCP1. So this confirmed two things. Number one is in the mature adipocytes, it's not present very early on, doesn't seem to contribute to the process of the decision of the differentiation, and appears when the brown fat is already a mature adipocyte and ready to function. So the involvement of BMP8 in the differentiation process was not something that we could suspect from this, and made this extremely different from the BMP7. And the next thing that we do is trying to validate physiology to see if the molecule that we are interested in follows the expected results with respect to the physiological response. So for this, we did two different challenges, high-fat diet and cold exposure. High-fat diet and cold exposure are two physiological conditions where brown fat gets activated. High-fat diet activates brown fat, but particularly cold exposure is the maximal stimulus that you could have there through activation of the sympathetic tone. And basically what you can see here is BMP8 behaved as expected. I mean, cold exposed took the expression of this to 120 times in the brown fat, extremely highly regulated. High-fat diet, again, was doing for BMP8 expected phenotype, was highly induced. However, for the other BMPs, what we found was that this regulation didn't happen, or in the context of the BMP7 or BMP4, we saw an induction that is compatible with their contribution to the differentiation of the brown fat, but is not compatible with just an acute response to the cold exposure. So that means that confirmed that BMP8 seemed to be in the brown fat, regulated as appropriately on the basis of the physiological regulation. The other thing is, when you fast, or when you decrease food intake, the normal response is to decrease energy expenditure. And then we reanalyzed the expression of all the BMPs on the concept of fasting and refeeding. So the expectation would be that something that contributes to thermogenesis should be downregulated by fasting, because you decrease your energy expenditure. and vice versa. That should be reactivated with the refeeding. And BMP8 was the only one that went down with the fasting and the only one that was going up with the refeeding in a very marked way. So after doing this, basically, it's something that we do to reinforce. I mean, if this quick, easy test fail, then the likelihood is that this molecule, if it's involved in this, will be much more complex with respect to how, or the function there. But that was very encouraging. The other thing is a pharmacological proof. If you treat with beta-3 agonists, these drugs are supposed to increase energy expenditure, activate brown fat, and we could show that BMP8 was induced by beta-3 adrenergic receptor. And the other thing that was important is, remember that we identified BMP8 because in our PPAR gamma models, they have an upregulation that we thought was trying to contribute to maintain thermogenesis. So if we treat the animals with PPAR gamma agonists so that we activate the PPAR gamma, what we find is the opposite effect. So that means if you don't have PPAR gamma, BMP8 increases. If we treat with an agonist of PPAR gamma in a model, we can repress the levels of BMP8. And this is a situation that contributes also to an anabolic state that facilitates fat deposition. Then we were looking at the promoters of BMP8 and BMP beta. So and the promoter of the BMP8 in different species. And what we found was two different types of response elements to begin with. As expected or not as expected, we confirmed that they have a thyroid response element, which suggests the thyroid hormone may be a regulator. We think that this is relevant. However, our impression was the thyroid hormone doesn't change in the way that is so acutely regulated. So may do it, but we don't think it's the main factor that determines the variability of the system or the highly regulated type of the system. And the other transcription factor we have found is PPAR alpha, or PPAR that will be relevant for PPAR alpha. We think that, in fact, it's very relevant for the thyroid hormone signaling through the TR beta 1, and we think that it's very more relevant for PPAR alpha than for PPAR gamma in the context of facilitating also oxidative processes in the brown fat. And then we did some physiology again. We like the physiology. We take some animals, we make them hypothyroid. In these animals, we see that the BMP8 is lower than in controls. And when we treat them with thyroid hormone, we can see the BMP8 can come alive. So you can see in vivo that thyroid hormone can regulate the levels of the protein in the brown fat. Then we start to work with a knockout mouse model. So this is a global knockout mouse. This mouse was published, and the phenotype that they had was a phenotype that was in the gonad of the males at the level of the Sertoli cells that made them reproductively not very competent. So that's the reason why people have never followed the animal from any point of view. So we got these animals, and we, at the beginning, we had many problems with the reproduction of the animal. We were very poor. However, we did something that really worked very well. We took the animals and we bred them. Instead of at 21 degrees, we increased the temperature to 24, 25 degrees, so that the animals were not that dependent on the brown fat. And we went from having a 10% viability in our, sorry, instead of 25%, 10, we went to 22% just by changing the temperature of the room, so that the brown fat was not that relevant in very early stages of the animal, so that they can compensate not having brown fat. The next thing that we observed was, these animals on a normal diet, they become obese compared to the knockout mouse, okay? And we will put them on a high-fat diet. As expected, the knockout mouse become even fatter. So that means if we suspect a defect in energy expenditure, it's not unexpected that the animals will become obese, and that when we stress them with high-fat diet, they become even more obese. However, there was something very interesting this year. Usually, you get obese, you will expect that the animals may eat more. But to have an animal that gets that obese eating less suggests that defects in energy expenditure may be quite robust. So again, it's a mouse, you remove BMP8, the animal becomes obese, they eat less, so we thought that the effect on the energy dissipation may be relevant here. That's proof of concept. Okay, and then we could confirm that these animals on a high-fat diet, the increase in body weight was at the expense of the adipose tissue, which suggests that they were in positive energy balance. In these animals, what we found is that they have, in fact, a decreased energy expenditure. That doesn't require to be a massive decrease in energy expenditure. this is sufficient to justify the phenotype from this point of view, okay? But keep in mind that there's this decrease in energy expenditure associated with an animal that eats less, okay? And then we learned when we did the gene expression analysis of the animal that what happened, I mean, here we have genes that are very much related to thermogenesis, PPARs, and lipid metabolism. What we found, and in these two lines represent to the mouse in chow diet, and these represent the animal on a high-fat diet. What we find is that the knockout mouse on a chow diet, they have an induction of the program of thermogenesis, in the sense that they, in the chow, they try to compensate, and they upregulate. But when we put them in the high-fat diet, what we find is that they are at maximal regulation. The wild type can reach this level, but the knockout doesn't go further because the system is upregulated under basal conditions, but cannot go beyond because it's blocked in their capacity. So this suggests that the brown fat of these animals is fully activated under normal conditions to the maximum capacity, but they cannot go further when we challenge them, and that's the reason why they separate in their body weight. Okay? Then we did another experiment, which consists, we take these animals, we put them in the cold for 3 weeks. So what they do when they're in the cold for 3 weeks is that they increase the brown fat. They just increase the amount of brown fat that they produce. Then at that moment, we inject them with norepinephrine, which is going to tell you the maximum capacity that the brown fat of these animals have to make heat. Okay? So the idea is put them for 4 degrees, 3 weeks, they develop the brown fat to the maximum, then you challenge to see how much heat they can make. So basically what we have here is animals that are 4 degrees and the animals that were at 30 degrees. In the groups of animals that we had at 4 degrees, our knockout mouse, what we found was that they have unimpaired capacity to reach maximal capacity. So the wild type could induce maximal response, but our knockout mouse have around 20% inability to reach their maximal capacity. So the lack of BMP8, in some way what they did is to prevent this brown fat from being able, not to make heat, but to reach maximal capacity for this. And in order to make heat, what is important in the brown fat is that the lipid droplets will activate lipolysis because they need to release the lipids to go to the mitochondria to be oxidized and to make heat. And what we found is that in the knockout mouse models, the defects of the BMP8 in the system, what we observed was that if we added BMP8 to the brown fat, we could see an increase in vitro of glycerol release. And glycerol is a parameter of lipolysis. So what we think is that BMP8 optimizes the production of heat through the activation of the lipolysis, allowing the fuel to go to the brown fat and being oxidized and making heat. And this is what you can measure with respect to the glycerol release. The other thing that we did in collaboration with a guy from Iowa is to measure electrophysiology, the innervation of the sympathetic tone into the brown fat. In order to make heat an animal, the experience cold, then the brain sends a signal to the sympathetic tone to the brown fat, and that makes the animal to make heat, correct? So what we found is you take the mouse, you let the mouse getting colder, that means the animal needs to make heat, and then they activate the sympathetic tone, correct? So, okay, what we have here is that as the wild type animal was getting colder, they could increase their firing rate of the neurons going to the brown fat, suggesting that they were trying to make heat. However, in our knockout mouse, what we found is that as the animal was losing temperature, the brain was sending an increase of firing rate to the brown fat saying, you need to work harder, we are not making enough heat. So that means the brown fat was defective in making heat. But that didn't mean that the brain was not trying to hit them very strongly to optimize their maximum capacity. Okay? And the other thing is, if we look at the brown fat of these animals under these conditions, and with an increase of the sympathetic tone, what we found is that some of the molecules that are involved in early stages of the signaling of the thermogenic response were actually more induced in the knockout, despite the fact that they were not able to make heat. So that means BMP8 was working somewhere below. You have an increased sympathetic tone getting in. The upper part of the signal transition pathway was upregulated but couldn't go through to make heat in these animals. So if you look at the signaling cascade of these animals, then what you have here is that, for example, the upper part of the cascade was more active, but when you look at the MAP kinase P38, for example, in our animals, on the activation of phosphorylation of phosphocrypt, in our animals there was a separation between the upper part of the signaling pathway and the lower part, depending whether you have BMP8 or not. The upper part was overactive, but there was a blockade below which the effectors that control the thermogenic response couldn't work appropriately. And then in vitro, we confirm that having BMP8 by itself didn't result in an increased thermogenesis, but having BMP8 made the response to norepinephrine amplify the response to norepinephrine, so that makes more sensitive the cells to the effect of the norepinephrine signal. So in this respect, we don't think that BMP8 by itself is able to create the signal. What we think is that BMP8 facilitates that the response of the system to norepinephrine is more effective. And we think this is being done through facilitation of the lipolytic response, allowing more fuel to go to the mitochondria. Okay. So then we have done a few different types of experiments looking at the readout of glycerol, showing that BMP8 increased the release of glycerol, as we said before. And then we were using a specific drugs that were blocking either alpha-2 receptors. And also we used one drug that was an antagonist of ALK7 that is one of the receptors for the BMPs. So we were trying to identify which was the receptor through which BMP-A beta was working. What we found is that if we were able to block ALK7, we could prevent the effects of BMP-8 on the lipolytic response. So that we think is BMP-8 effect could be mediated. We have pharmacological support for this effect. But then we were really bothered by the fact that the animal was eating less, and the fact that BMP8 was also in the brain, and L7 actually is also in the brain. And then we look at the neuropeptide expression in the hypothalamus of an animal that was eating less. And we were very positively surprised because these animals have a decrease of the orexigenic neuropeptides and an increase of the anorexigenic peptides. And this is very robust. Even when we did that, there are many other models where this type of response is not that strong. So I think there is no doubt that this animal is eating less, actively eating less with the proper changes in the neuropeptides. And then we went back to some of the work that we have done before looking at the effects of thyroid hormone in the hypothalamus and the effects in activation of brown fat. That was our previous paper. And we showed that the brown fat, for example, when we put ICB, thyroid hormone, in the brain, that increases the activation of the sympathetic tone and the activation of the brown fat, and this is dose dependent. And if we injected this specifically in the ventromedial hypothalamus, we can recapitulate the effect of the thyroid hormone at the level of the ventromedial hypothalamus, signaling to the brown fat. And as you can see here, not here, but and in this context, what we know is that the thyroid hormone does that through downregulation of AMP kinase in the brain, and that was as a result of the previous link. So the concept is that if you inhibit AMP kinase in the hypothalamus, this is associated with an activation of the brown fat. So what happened when you put BMP8 in the brain of these animals? Okay, one of the things that we observed when we put these animals ICB is something very important. So we injected in the third ventricle BMP8 or control, and we had as a readout the firing rate of the sympathetic tone in the brown fat. And we were comparing the brown fat with the kidney. One of the key problems of the sympathetic outflow is that you can go to the brown fat, but you can go to the kidney, or you can go to the heart. But by putting BMP8 in the brain, we could get an activation of the sympathetic tone very specifically going to the brown fat, but not going to the kidney. This is very important because most of the drugs that increase the sympathetic tone to decrease body weight, they help you to lose weight because you may activate the brown fat, but they kill you because the activation of the sympathetic tone is not specific enough and goes to the kidney and goes to the heart. But by introducing BMP8 in the brain, we could get a specific activation of the sympathetic tone in the brown fat without being affected the kidney. And from the point of view of the future development of this opens the possibility of nucleus-specific regulation of the sympathetic tone to specific organs, in the sense that you could get approaches that may target the brown fat without touching organs that you don't want. And these animals, when we did that, they decrease their body weight and they maintain it as a result of the activation of the brown fat. But we didn't see a link between this and the food intake. So that was very specific intervention that was targeting energy expenditure, but not food intake in this model. The other thing that we looked at is what were the effects of the BMP8 in the hypothalamus activating C4. C4 is a marker of neuron activation. And what this result shows is that if we inject BMP8, we can get force activation in the neurons that are injected with this. But when we use the inhibitor of ALK7, the receptor that we thought was for BMP8, we could prevent or reverse the response of the activation of C4, further suggesting that ALK7 receptor may be the receptor mediating the effect of BMP8 in the hypothalamus. Interestingly, during all these experiments, we could see that BMP8 treatment in the brain contributed to increase the temperature in the animal. So there is an effect, a defect of the whole temperature. At the same time, injecting BMP8 in the brain makes these animals leaner. So you activate the brown fat, and they spend more energy, and they lose weight. And we could see that at the molecular level, in the brown fat of these animals, putting BMP8 in the brain, we could get an activation of the brown fat at molecular level too. What I said before is that the brown fat is not only useful for you to lose weight, but also to clear fat from other organs and to improve insulin sensitivity. And one thing that was very interesting is the fact that these animals have, when you inject in the brain, they have a tendency of glucose to go down. Triglycerides were going down. And most importantly, and I don't have it here, the insulin levels of these animals were lower. So you have a tendency to decrease glucose, and you have lower levels of insulin. That equals to increased insulin sensitivity. So you can show that putting BMP8 in the brain, activated brown fat, make these animals to be able to control their glucose better, and they needed less insulin because they were more sensitive, or they could use probably glucose through the brown fat, and they didn't need insulin to do it. So in this respect, what the results showed before from the thyroid hormone is that inactivation of AMPK is the one that is associated with an increase in brown fat. So we wanted to know if BMP8A in the brain was able to inhibit AMPK and contribute to the signaling to the brown fat. However, from our experience, what we know is that if we inhibit AMPK in the BMN using dominant negative viruses of AMPK, we can get an induction of markers of brown fat in the brown fat. Okay? Or brown fat activations. I mean, inhibition of AMPK in the hypothalamus results in activation of the sympathetic tone and activation of the brown fat. And this is an effect that we can block with beta-3 beta blockers. So that means goes to the brain, activates the brown fat, but if we give the animal beta-3 beta blockers, we can prevent the effect of the innervation in the brown fat directly at peripheral level. So we know it's mediated through the activation of the adrenergic system. However, when we injected the animals in the brain with BMP8, we didn't see the expected decrease in AMP kinase activity. We actually saw the opposite effect. So initially, we were not sure. I mean, initially we thought that as a disappointment, but in some way, as I say, BMP8 increased the phosphorylation of AMP kinase. We have seen what happened before with thyroid hormone. If we do this in the cold, again, the effect of the cold is a decrease of AMPK phosphorylation and activation. But BMP8 was doing the opposite in the AMPK pathway. So that, at that moment, caused a problem for us. However, further experiments, what we could demonstrate is that BMP8 was acting below, in the cascade of AMPK, but below EMPK. So that's the reason why we didn't see it. And that the upregulation of AMPK in the model is probably an adaptive mechanism after the cascade down has been activated. And we could demonstrate this by using viruses that were dominant negative or constitutive active in the brain, plus minus BMP8. And we could show that BMP8 could induce the expression in the presence of the AMP kinase dominant negative. the bottom line of this is we think at the present moment that BMPA works at the level of the brain, that knockdown of AMPK facilitates thermogenesis, but if you have BMP8, facilitates the cascade of this and may have a retroactive feedback that may, under these conditions, even increase AMPK. So we have been able to locate where BMP8 in the brain should interfere with the AMPK signal that happen below in the signal transduction pathway. The other thing that we did is to use thermoimaging of the brown fat of these animals. And here, for example, you have animals with the GFP, the AMP kinase dominant negative, and the AMP kinase constitutive active. And here, what we have is that when we inject the animals with BMP8, the thermogenic response in the area where the brown fat is much more increased. We can see that the brown fat is there and makes more heat, and we can detect it with a thermal camera on top of it. And that this effect is reversed by AMP kinase constitutive active in the brain. And when we did a correlation between the changes in the temperature and the changes in the bath temperature, what we can see is that there's a correlation between the temperature in the brown fat and the temperature in the animal. To suggest that really the contribution of the brown fat at this regulation of BMP8 to the thermogenesis and temperature is directly or more specifically mediated through the brown fat of these animals. So the model that we have here, we have thyroid hormone inhibiting AMPK or cold, inducing de novo lipogenesis and facilitating brown fat activation. BMP8 follows the same path, but is situated below AMP kinase and can interfere with it. So basically, in this paper, what we show is that BMP8 is different. It's not making brown fat to be more abundant. What it does is to be a sensitizer of the adrenergic stimulation in the brown fat that makes that with less sympathetic inflow, you can have an amplified response. And this is very specific of the brown fat because BMP8 is very specific of the brown fat itself. So you could have potentially systems that you can sensitize the brown fat to the sympathetic tone so that with less activation you get the response that you want. At the same time, that suggests that you could use this type of, our next step is to try to characterize the signaling pathway below this, because if we could target some of the downstream events, specifically in the brown fat, that will allow us to target the brown fat without touching the other organs. There's one thing which we will exploit in the future, and is that we think there is a connection between the brown fat and heat production and the sympathetic tone in the brain. So if you are cold, you activate the sympathetic tone and you try to make more heat. But if you are warm and you have too much heat, what we have seen in some of the models that we have is that the brown fat seems to be less active. And we think it's because there is some kind of feedback going to the brain and decreasing the sympathetic tone to the system. So the challenge is, if we find a way how to increase the sensitivity in the brown fat to the adrenergic signal, we make the brown fat to be more competent at making heat. We may have the added beneficial effect that the sympathetic outflow from the brain may not be that, or may even be decreased. So that we may get a beneficial knock-on effect on the sympathetic activation effects in heart or in kidney. In summary, I think you have the benefits of activating brown fat, the benefits of not activating the other organs, and the potential benefit of, by doing so, even shutting down some of the sympathetic activation in organs that are more negative. If we think in obesity, we know that strategies that increase energy expenditure make people to lose weight. The problem of these strategies, I'm thinking in fen-phen or sibutramine, have been that by doing so, by activating the sympathetic tone, they could get people to lose weight. But at the same time, these individuals have cardiovascular problems. So in this regard, if we uncouple the effects from the sympathetic tone to the brown fat or other organs, that could be an approach that could be safe and at the same time make people to lose weight. The second implication from this is that potentially we can go to the brain and map the neurons and the nuclei that control the sympathetic flow to specific organs. And this is what we will be doing as a second part of it by doing two different types of things, which is nucleus-specific knockdown of BMPA in the brain, different regions, trying to see the different outputs to different organs, and tracing experiments from the brown fat to try to see which type of regions get infected, and try to compare this with, for example, models of obesity and metabolic syndrome, where we know that the sympathetic outflow goes not only to the brown fat, but also to other organs like cardiovascular ones. And the concept that we are trying to portray is to try to separate or to identify areas that are mainly controlling the brown fat. And to another level will be to try to determine if there is anything specific in these two different types of neurons that may help to understand why they may get activated by two different types of responses. We know that people, when they are exposed to cold, they increase the sympathetic tone. In vivo, we know that the brown fat of these individuals get activated. We know that the sympathetic tone to the heart is activated, but this is sort of like a healthy sympathetic tone, because what it does is the heart becomes stronger, become more competent to deal with excess. However, this is different from the response that we see in an obese individual, and they have an increased sympathetic activation that goes to the brown fat somehow, but particularly to the kidney and to the heart. And I think these two models suggest that sympathetic tone could be good, but not all of them. And ideally, if you could separate this centrally, or if you at some point could facilitate mechanisms like the BMP8 in periphery to make it more sensitive to the sympathetic inflow, that could separate the two components of it. The key people in this project has been Andy, particularly, and Stefania, who is here. And this project has been a few years, more years than we should have done, but the problem is that it was very intense and that people was very generous, and we have like 3 people that were really the main drivers of this. The main problem sometimes is that you need to have a team of people to address big questions. If you look at things with a small vision, then, and you want to have your own project and be an isolated island and from beginning to the end, it's very difficult. And in some way, part of my job has been to try to make sure that everyone was happy through the process and everyone get the credit for this. And they feel that they have contributed a job that I think is probably one of the best in my laboratory. So this is what I want to say, my collaborators, colleagues, industry support and funding agencies, and thank you for your attendance.
Have you gotten around to looking at patients to see if obese people have SNP changes, mutations in BMP?
Yes.
HTP?
We have been working with the people from epidemiology. The initial data suggested there may be something there. However, it's very difficult because the type of population that you need to choose with respect to try to find a genetic modification, they need to have a specific type of phenotype that may be obese, but we need to find people with a preferential defect in energy dissipation. And many of the people or collections that these type of phenotypes are not extremely well established. So the link is relatively weak, but it's there. The other thing that we are doing is trying to develop a good antibody that works in blood from these individuals, because we think this works at the level of the brown fat, but I would not be surprised if this could be released into blood and also have some kind of endocrine role. Or simply, if we can use it as a marker of brown fat activity there. So that's the part that we are trying to do. The other thing is that from the point of view of treatments, this is a type of molecule that could be, we need to do, we're working now with a transgenic mouse. We know that these animals, they don't get leaner, but they get much more insulin sensitive. And we're working with a lean model at the present moment. So in a C57 that we are back crossing the transgenic mouse there, we think that we will see an effect in body weight. But the impact on metabolism is much more evident at these early stages. So I'm not that worried whether this is the cause or not of obesity. I'm more positive about the possibility that you could use it as a therapeutic approach. And particularly, I think for us what is very interesting is the concept that you can use a sensitizer for adrenergic stimulation. up till now we have been always working on the assumption that you put the norepinephrine and then the whole cascade goes. The fact that you can have a system that can be induced when it decompensates, and what it does is to optimize the signaling, that opens opportunity to find this type of mechanism and trying to work on them and whereas given molecules that could have a broad spectrum may be a little more difficult, you just target this, I think that will be very safe.
Any other?
I don't know if you are exhausted, uninterested, or you haven't understood anything. That's the three options I can think of.
Yeah, thinking about the influence of the temperature, these experiments, I was just thinking, is there more obese people in, let's say, countries, more warmer countries, and is there maybe some kind of effect of their BMP8 there, like that is more active? How does it work between regions in the world?
Okay, there is some epidemiological suggestion that there is a gradient north to south for obesity. However, the confounding factors are tremendous because the north, it's not that you are exposed to cold in the north. I mean, people there don't like the cold, and even, but despite this, you can see a trend that may be supportive of it, but you need to consider that all these people are not naked in their own countries trying to go through the weather. But there is some suggestion that this may be true. And you see the Greek thing, that's quite supportive of the whole thing. But I mean, I think I cannot say something very rational on this.
More? Is the mice, the knockout mice, you said is expressed, well, I don't know, this BMP is expressed in brain and brown fat. But during development, I mean, the embryos and in development, is it expressed in other tissues, or is always this pattern?
We haven't done studies in the embryos. I know that they develop, if they are born and we put them at higher temperatures during the early stages, and then they survive the first week, they go normal.
Yeah, but in wild type, is it expressed only in these two tissues, or in embryos is also expressed in any other tissue?
The thing is that we haven't done studies of the development of the embryos to know when and where they appear. I mean, we haven't done that. Yeah, so that's the answer to this. I think at the beginning we were a little worried because the first report on this suggested that the animals have problems with reproduction. And then we were worried about the hormonal component of it. But we checked in these animals that their sexual hormones were normal all the way. The endocrine cells of the gonads are not affected. What they have affected are the cells that control the ovules and the sperm, but not the endocrine component. So I think there's no differences in things like estrogens that may have had a confounded factor. There are no differences in the bone, suggesting any type. No, I mean, this BMP8 is quite safe with respect to effects in the bone. And the other thing that we were thinking is if these animals had been born with, let's say, differences in body weight early on, whether any type of programming effect may have affected this. But when they are born, they are not different in size to suggest any type of malnutrition intrautero or any defect there. So they are born equal. The only thing is that when they die during the first 3 to 4 days, which is the moment when the animal has less fat, is less thermoinsulated, they have a major burst of sympathetic activation trying to make heat, and a failure to make heat at that moment is really lethal. So that's the reason why we put them in the warm, and we could rescue the phenotype that give you a sense that the temperature was a key factor in the animals that was challenging their survival. And if you stop that, I would say that the other place where BMP8 is present is in the gut, and that's what we are following now.
Do you see changes in lipid metabolism, overall lipid metabolism, like if you do lipidomics of serum? Do you see in a more oxidated fatty acids or plasmalogy? I mean, is there any change?
We haven't done a proper profiling of lipids in these animals because we want to focus more on the transgenic, which is the one that is going to be healthy. And then it's the one in which we will prioritize trying to confirm if overactivation of the brown fat in these animals helps them to have a better lipid profile that will be a little healthier. From the point of view of the beast one, we have reached to where we have reached until next week, and now we are going to the next phase, which is trying to determine what is going to be the different directions that we're going. And I think, I said, the top priority we have is the brain, try to map it. And the other thing will be to try to find molecular mechanisms regulated by BMP8 in brown fat that may help us to maybe identify something that's more specific or different from what we know. We don't know how BMP8 signal really interferes with this. And because we have a blockade, we can see the upper part being overactive, so we know more or less where to start looking for the interaction. So that will be the top priorities. The human aspect that you mentioned, which is definitely very interesting, we are going to do this through we need an assay to measure it. The other thing, we have an active program now where we are looking at the effects of cold exposure in humans and the effect of a specific type of nutrients on the activity of the brown fat. And paradoxically, some of these interventions in human at the level of transcript, we know that BMP8 is in human brown fat, and it's appropriately regulated by cold in the brown fat of a human. But we have not been able to detect the protein in humans.
But mice also lose brown fat very rapidly after birth?
Well, it's very interesting because in the transgenic mouse, at the beginning, you get brown fat in the transgenic period. As the animal puts on weight and becomes more thermoisolated, we lose the expression of BMP8. But when we put them on a high-fat diet, which means that increases the sympathetic tone and reappears again the high expression. So that suggests that during the evolution of this, as the animal is putting more fat inside or around, then definitely they need to make less heat, and then the activation of the brown fat becomes less. And then you have different, that's something which is very relevant with respect to the strain of the mice. If you go for a 129, what we know is that the 129 probably doesn't become obese because they have more brown fat in the brown, in the muscle, and even in the white adipose tissue. And if you challenge the 129 with overfeeding, the animal resists the obesity, but you can see an induction of the program that suggests an increase in sympathetic. And BMP8 is higher in a 129 than in a C57. That suggests that this model or this type of mechanism is relevant in the rodent. And even if you look at lipolysis in a C57 and a 129, in a 129 the lipolysis is much tightly regulated and much better control. In the C57 is being lost very quickly. So I don't know, this evidence suggests that this type of alterations in the sympathetic nervous system mediated activation of brown fat may be something that is differential between being prone to obesity or not. And the other thing is aging. We know as the animal gets older, the sympathetic tone gets older too. That means the tone is going down. So you combine obesity with age, that really has a severe effect on the sympathetic activation.
Any other comment, question? If not, thank you very much.
Okay.